Galarza. George, would you like to share your presentation? George will talk today about the effect of plant-based, animal-based, and algae complemented feed sources in vannamei and gut microbiota population. Go ahead, George.
All right, thank you, Dr. Delgado. Let me see. All right. So hope everyone is doing good. Hope everyone's staying safe. So my name is Jorge Galarza. I am a PhD student in the Department of Biology. And like Dr. Delgado mentioned, that is the title of my presentation. But today I will not be talking and implementing the supplementation of algae because I'm working with another student on getting those results analyzed. So first, an introduction to the vannamei shrimp. We're going to talk about the background, why it's important, why are we even doing this, the feed production, the analysis, and the results for a previous experiment that we conducted that led us to where we are now in this whole project. All right, so just a small background. If you have no idea about what a vannamei is a white-legged shrimp. Is the most popular shrimp that you could buy commercially. So you can find it in pretty much any supermarket in the US and around the world. If they sell shrimp, you're probably gonna buy some kind of vannamei, Latopenaeus vannamei. It's native to the coast of Sonora, Mexico, and it goes all the way down to the coast of Peru. It can grow to sizes of 23 centimeters or 9.1 inches, which it's pretty big. You know, it's a really big species of shrimp, even though there's a bigger one, but this is the mainly consumed one and farmed. So females are almost always going to be bigger than males in size and weight, of course. And yeah, so that was a little bit of a short background on the vannamei. Now we're going to talk about the importance of the vannamei and the importance of why we're making this this project actually happen. The vannamei, like I said, is the most important shrimp species in the world regarding commercial value, okay? And this is kind of verbatim. The shrimp market was valued at $45 billion US in 2019 and is predicted to increase 5.2% by 2024, okay? So that's a pretty hefty amount of growth from 2019 to 2024 as it's predicted. So why are we dealing with the feed? Why do we think it's a good idea to invest time and money and effort into producing a new feed? Well, the answer is because feeds are the largest costs in any shrimp aquaculture operation or any shrimp aquaculture farm, because the feeds, the expenses that go into the feed is actually 60% of the actual expenses of these aquaculture farms, okay? So the main ingredients in a lot of the feeds that they are feeding the shrimp right now is actually fish meal. And as you guys can see, 1 metric ton of fish meal is $1,480 US dollars. And a viable option, which we found out to be, is glandless cottonseed meal, which is a waste product that contains high fat and protein content, which is perfect to replace the fish meal. And not only that, but it's a waste product. So that means that it's a lot cheaper. So the total expenses that would go to the actual feed would reduce dramatically. So now GCSM, or glandless cottonseed meal, is $300 US dollars for a metric ton. Okay, so you guys can see the huge difference in prices. And one of them is a waste product and one of them is truly not. Okay, so following up on that, we're going to talk about the feed production. So in the first part of my experiment, this is a previous experiment that we conducted, we had 5 diets that we were trying to compare within each other. Okay. We had diet 1, or diet with 44% glandless cut and seed meal, 0% fish meal, which this diet on the left side of the table 1 has, as you can see, nothing coming from a animal source except the fish oil. Okay. But if you guys look at the table, you guys can see that I put in gray the sections that change in concentration of the ingredients that go into the feed. So diet 1 contains 44% glandless cut and seed meal, 0% fish meal. Diet 2, 33% glandless cut and seed meal, 11% fish meal, 22% glandless cut and seed meal, 22% fish meal. And then the last one is 11% glandless cut and seed meal and 33% fish meal. And then as our control, we use the commercial diet, the commercial feed. Okay. So now we didn't make the commercial feed. We bought the commercial feed as a control, correct? So the first 4 diets that we're going to talk about, we actually made them at school. We controlled every single ingredient. We controlled the parameters of making the feed, and we measured everything that we were doing. Okay. So how did we make the feed itself? We used the extrusion. We used an extruder that we have at NMSU, and extrusion is a process by which a set of mixed ingredients are forced through an opening. Okay. So the parameters that we used for the extrusion process is we used a 1-to-1 screw ratio, which means that inside of the extruder, we have this, this screw in the middle that, as you guys can see, in the beginning is thin, right? And then at the end of the extrusion process, it gets thicker, right? That creates more pressure inside of the extruder itself. But we didn't really want there to be a huge dramatic pressure change from inside environment of the extruder to the outside pressure, which is atmospheric pressure. So we didn't want the feed to expand too much, because then it was going to float a lot. And since shrimp are bottom feeders, we wanted the feed to be as compact as possible, so it could go to the bottom and sink. Okay. And that's one of the things that we measured through this experiment. So we used a 1-to-1 screw ratio, which means that the thickness at the beginning of the screw is the same thickness at the end. Okay, so we're not adding a lot of extra pressure. We had the screw rotating at 180 revolutions per minute. We had HZ1 set at 90, which HZ stands for hot zone 1, 2, and 3. So as you guys can see, these go in an incrementing level from 90 to 110 to 130. So hot zone 1, so you guys can get an idea, is let's call this a heat zone, right? So it would be heat zone 1, and then in the middle, it would be heat zone 2, and then at the end, it would be heat zone 3. So as you guys can see, as the screw is rotating, you're pushing out the powder that contains water, and it contains all of these ingredients in it. So when it comes out from the die, it's gone through a cooking process, right? And then at the end, you get your final result, which is your final feed for the shrimp. Okay, so how did we measure if the feed even worked, right? Because we can make a Cheeto and say, oh, it's kind of a Cheeto, but it's really not. Okay, so we made it to where every single diet that we created was measured by doing analyses on different physical chemical properties. And then we also did vannamei analyses, which, we measured all of these. So when we finished extruding, we measured ash content, crude fat, crude protein, digestible energy, metabolizable energy, expansion ratio, floatability index, hardness, scanning electron microscopy, water absorption, water activity. And from those, we compared it to the control, right? I know it's a mouthful, and I just read those from the list. But we had to do all of those to determine, is this even comparable, physical, chemically speaking? Is it about the same? Does it look the same? Does it behave the same? Does it have the same shelf life, for example? Does it have the same water activity, ash content, which is mineral content, crude fat, crude protein, and so on and so forth? So when we determined, okay, these feeds are a suitable option to replace feeds that only contain or mostly contain fish meal, then we went on to the second stage, which is vannamei analyses, which we conducted an in vivo growth and we conducted an in vivo digestibility. In vitro digestibility, so for the in vivo growth, we grew the shrimp for 30 days and then we measured the growth in those 30 days. And then for the in vitro digestibility, we just used two methods. We used the pH stat method and the pH drop method. And the following slide will show the results for that. So according to the in vitro digestibility, there was no significant difference in digestibility between feeds, okay? So what does that mean? That the hepatopancreas inside of the shrimp was able to digest statistically equally, all of the feeds. So all of the feeds were digested exactly with the same ability according to the pH drop method. So according to the in vivo growth, there was significant difference between total growth in the 30 days of growth between the 5 measurements, which is the 5 diets or the 5 feeds. And this is the interesting part. Results indicate that the commercial was able to reach a weight of 0.02 grams higher than the feeds we developed at NMSU, okay? So you get a 0.02 grams higher weight from the commercial feed in the shrimp than if you do it with the diets that we made at NMSU, okay? So from there, a lot of questions began to pop up. And 2 of them was, how does the change of the main protein and fat source in the feeds change the gut microbiota in the vannamei? And is it making the vannamei healthier, resulting in higher yield numbers in farms? So this is something that we will see in further results, but we will go ahead and move on to the second stage of my my project. And I don't know how I'm doing on time.
You're good. You still have 5 minutes, George.
Okay, sounds good. So we're going to talk about the gut microbiota and the gut microbiota background information, microbiota importance, methodology, and the expected results. Okay, so a background on what a microbiota is a collection of microbial communities colonizing a particular habitat, which in this case, we're only focusing in the gut, which means we're talking about the stomach, we're talking about the hepatopancreas, and we're talking about the intestines. That's it. All the microorganisms found in an environment, including bacteria, viruses, fungi, and archaea. As well, in this example, we're only focusing on bacteria, okay? So these bacteria, why are we even doing bacteria, right? These bacteria play crucial roles in nutrient absorption, water quality, which we measured, and the results are kind of intense, pathogen defense, antibiotic resistance, and maintaining host health in general, okay? So it is, the microbiota of the shrimp has been recognized as an endocrine organ, as a virtual endocrine organ inside and outside of the specimen itself. So under that, you can see the overall gut microbiota structures when analyzing the 16S-rRNA gene according to Huang et al. In 2018 from a saltwater natural habitat population, and the sample number was 3, okay? So what I put in the bottom is just what I'm trying to do, but with 4 different diets. And we'll talk about the diets shortly. So why is it important that we measure the microbiota of the gut in the shrimp, right? So since there is a decline in wild-caught shrimp and an increase in aquaculture practices, it's almost linear what you see is happening with the disease outbreaks and the frequency that they happen at. So with the increase of inland aquaculture practices, the disease outbreak frequencies has also increased, you know? So we go from wild caught, which is low, to inland farming, and then you have the frequencies of disease outbreaks also at a linear increase, okay? So like I said, these bacteria play a crucial role. And according to Clark et al. 2014, the gut microbiota has been recognized as an external and internal endocrine organ by acting as a pathogen barrier, providing metabolic pathways. And also it is important because it can be used to make a logical human health and diet connection, okay? Which this point steps outside of the shrimp topic. So for the methodology, just as before, we have the diets, and they're very similar to the diets that I talked about at the beginning. But this one is 44% glandless cottonseed meal, 0% fish meal. Half of it is glandless cottonseed meal, half of it is fish meal, and then 0% glandless cottonseed meal, 44% fish meal, and then we have the control diet, which is a commercial diet. Okay, so in order to do this, we have to conduct a growth for 30 days with the different feeds, which in the bottom, you will see the parameters of everything that we have done. Then we dissect the gut of the vannamei and put in rRNA DNA shield, then store it at -20 until it is used.
1 minute, George, please. 1 more minute.
And then we extract the microbial DNA, and then using primers 338 forward and 806 reverse, we use it to amplify the V3 to V4 hypervariable region of the 16S rRNA gene. Then we conduct the PCR reaction, and then we do the Illumina MySeq sequencing. And then after that, the last step is data analysis, okay? Which that, even though is the smallest cell in this slide, it is actually the hardest one to do. So the expected results, according to many articles, everything changes the gut microbiota in the vannamei. You're talking about water pH, water salinity, water temperature, the parameters of ammonia, nitrite, nitrate, the sediment, and the feed. Okay, so according to Sheng et al., the simple supplementation of shrimp feed with soy protein significantly reduces the abundance of potential pathogens, such as Vibrio, which is a big deal, and Flavobacterium in the shrimp's gut. Okay, so it can be predicted that a substantial or change in microbial communities in the gut of the vannamei will be observed by the end of the experiment. And that's it. Thank you.
Okay, thank you, George, for your presentation. We have only time for one question. Who would like to ask George? No question, then I just have one question, George, really quick. Yes. How many academics does it take to change a light bulb?
Hopefully one.
The answer is none. That's what their students are for. Okay, thank you guys. If you please help me give George an applause for his presentation.